It's Miss Barnhart, and today we're going to begin our study of genetics. The father of genetics was an Austrian monk by the name of Gregor Mendel, who lived in the mid 1800s. At the time, the current theory for inheritance was the blending theory. This diagram illustrates what was thought to occur. Both parents contributed equally to the trait of the offspring. Makes sense, right? Actually, no. If this were the case, the traits would be diluted over time. It also doesn't help explain how traits can skip generations over time. So, if Mendel were interested in studying genetics, one question is, why peas? Peas are an example of what is known as a model organism, one that is useful for studying traits. So what's so great about peas? First, they are easy to grow and maintain. It's pretty easy to control their mating since they usually self-pollinate. They produce lots of offspring, seeds, and their traits are conspicuous. And they have a short generation time. Let's take a look at Mendel's method. He established true breeding lines, crossed those lines using cross-pollination, and then counted the offspring. Mendel studied 7 different distinct traits. For any trait, when 2 different lines are crossed, the first generation, the F1, only showed one of the traits. When 2 of the F1 are allowed to self-pollinate, both of the traits appear in the F2. And they always appeared in a 3-to-1 ratio. When he crossed round and wrinkled seeds, all round seeds appeared in the F1, and 5,474 to 850 in the F2. Yellow seeds were dominant to green seeds, 6,022 with yellow to 2,001 in the F2. Purple flowers dominated over white flowers, 705 to 224. Inflated pods were the dominant trait over constricted pods, 882 to 299 in the second generation. Green pea pods all appeared in the first generation, 428 to 152 green to yellow in the F2. Flowers appearing at the sides, called axial, were dominant over flowers appearing in the terminal. 607 showed axial flowers to 207 terminal. Tall plants dominated over short plants in the F1, with 787 to 277 ratio in the F2. From these results, Mendel concluded that traits exist as 2 varieties. We now call these forms alleles. And each individual possessed 2 alleles for each trait. One from their mom and one from their dad. It was the combination of these traits that resulted in the physical appearance of the offspring. While Mendel was not aware of chromosomes, we can apply his findings to our current understandings. Each individual has 2 types of each type of chromosome. Remember, these are called homologous chromosomes. One chromosome will be inherited from mom, the other from dad. The bars shown here are representative of alleles. Notice that each chromosome has a colored bar at the same location or locus. The individual shown here has 1 allele for purple flowers and 1 allele for white flowers. Before we continue, let's define some helpful genetic terms. If you cannot readily state what each of these terms mean, please pause the video to record the definitions. These are crucial for your success in this unit. Trait. A trait is an inherited characteristic. A gene is a sequence of DNA bases that code for a protein which is expressed as a trait. Each gene has different forms or alleles. An allele that always expresses itself is said to be the dominant allele, where one that can be masked by the presence of the dominant one is the recessive allele. Each individual will have two alleles for each trait. The genetic combination of the alleles is the genotype. If the allele pair is identical, we say the individual has a homozygous genotype. This could be either homozygous dominant or homozygous recessive. If the allele pair is different, in other words, one dominant and one recessive allele, then we say the genotype is heterozygous. We call this physical expression the phenotype. We will use capital letters to represent the dominant allele and lowercase letters to represent the recessive allele. I highly recommend that you select letters that you can clearly distinguish uppercase from lowercase, because this will make it much easier for you to distinguish them as you work through the remainder of the module. If I use the letter A, the capital A represents the dominant allele, while little a represents the recessive allele. Big A represents the homozygous dominant. Big A represents the heterozygous. And little a represents the homozygous recessive. Note that big A and big A will have the dominant phenotype, while little a will express the recessive phenotype. Mendel developed two laws of inheritance from these investigations. First, the law of segregation states that only one of each allele pair goes into a gamete. This is a chance event, much like flipping a coin or rolling dice. In this case, there's a 1 in 2 chance of each allele ending up in the gamete. The law of independent assortment states that separate traits are passed on independently of each other. This is provided the genes are on separate chromosomes, not linked. Both laws can be explained with the behavior of chromosomes during meiosis. The alignment of the paired chromosomes during metaphase 1 is totally random. And during the anaphases, the alleles separate from each other. The law of independent assortment explains why combination of traits not present in the parents can end up in the offspring. This led to the death of the blending hypothesis. I hope that's helpful.